Alright, hello everybody. Welcome back. In this series of videos, I'm going to be reading the AlphaFold paper, which is this paper right here, by DeepMind, that won the Nobel Prize. And I don't have any background in biochemistry whatsoever, right? So I'm acquiring the knowledge as I go along using a tool called DenseWiki, uh, which I'm the founder of actually. But you don't have to use DenseWiki to read this paper unless you want to. And yeah, so let's read along. In the last section, in the last live stream, I went over the abstract, which took a lot of time because I had to learn the basic concepts of what's going on. Uh, and we went through the main section where we basically understood that the AlphaFold network is a league above in accuracy compared to previous methods, and we saw both the um, quantitative as well as somewhat the qualitative proof. We saw that the accuracy is 3 times better, and also it also looks more accurate, at least to the naked eye, when it's described in this way. We haven't yet gone into the details of how the network works, how this thing works, which I think we will go into today. Um, but yeah, that's kind of what where we're going. We left off here in this paragraph, and where I was trying to understand what this global superposition metric template modeling score means. Um, and This is a score which basically assesses the structural similarity between two protein structures, right? And they were able to predict this score as well, predicted this estimate, and used that as a confidence measure, basically. I think. Wait, we also find that the global climate actually estimated. We overall this. Yeah. So, but no, not as a confidence measure, but like they used they being able to predict this modeling score using the network was itself a proof of the accuracy of the network, right? If you know, if you're really good at taking tests, if you're so good that you can even predict your score on the test in advance, that is incredible, right? If you can score yourself really well. So anyway, this figure, I let's go into this one step by step. The analyzed structures are newer than any structure in the training set. Okay, fair enough. Which means basically that the validation set was new, right? For the filtering is applied to reduce redundancy. See methods. Okay, we'll get to that later. Histogram of backbone RMSD. Backbone RMSD, again, backbone, as we learned last time, is that protein structures have a main chain, the backbone, and then they have all these side chains. So um, the back if you just look at the root mean square deviation for the backbone structures, that's one metric, and then there's other metrics for the side chains and everything, right? And then the auxiliary atoms RMSD, these are the main carbon atoms in the chain, right? um, and this is for the angles. So the backbone RMSD is for the angles in the chain, and I think this is for the actual positions of the carbon atoms, if I remember correctly, right? Let's quickly look at the ARMSD protein backbone accuracy, right? Yeah, backbone structure, right? And then this was for the um, let's see, the major angles between, yeah. So the backbone accuracy focuses on the angles between the links, right? By the way, am I sharing the right screen or not? Okay, I'm sharing the right screen. And then the I think we look at the aux carbon, right? The carbon, alpha carbon atoms. This was for the actual positions of the carbon atoms, right? Okay, error bars are, this is like figure A. Where's figure A? This one, right? So, uh, histogram for full chains. Uh, full chain RMST. This is the, the score that we're looking at. The angstrom RMST. Angstrom is the distance, right? So this is about how close they are to the distance. The actual atoms and the predicted positions of the atoms, how close they are, and the lower the better, right? So 0 to 95% confidence intervals. Fraction of protein. Okay, most proteins were within a zero point 0.5 to 1 angstrom distance, and a few proteins were really really good, like really really accurate, and most were like in this way. Quite a bit of, although like, okay, this is interesting, quite a bit of proteins were also like more than 8 angstroms, which is not very accurate. So it seems like it does really well on the majority in a way of protein structures, but also there's a lot of structures that it's not able to predict at all in a way, right? uh, okay, now this is the fraction of correct x1 rotamers. Let's look at what that is. The data excludes proteins with a template. Search. Okay, I'm gonna look it up what this means. What does this data set exclude, right? And by the way, let me set a timer for like 30 minutes ish, something like that, so that we don't go too crazy. Okay. Search, a software tool used to search protein or DNA sequences against a profile hidden Markov model database. Okay. So the main concepts here, if you haven't used density before, the main concepts to understand this are search itself, and then hidden Markov model. What that is, that's important to understand, and then sequence identity. Let's look at what this is first. I already know what this is. Root mean square deviation. Where a template is a known protein structure used as a guide for predicting or modeling the structure of a related protein. Okay, I guess that makes sense. Then a sequence identity. Let's actually first take a look at what this template means. Okay, a template is a known structure of a protein that is similar to the protein you're trying to understand. If you want to build a new house but don't have plans, you might look at existing houses that are similar and use their plans as a starting point. Okay. Figuring out a protein structure directly can be very expensive. Using a template helps you quickly predict. Okay, makes sense. Okay, now let's look at what is a sequence identity. Sequence identity in bioinformatics is like comparing two sentences to see how many words are the same and in the same order. Okay, how similar are two protein structures? Okay, if two proteins have 90% sequence identity, they probably do similar jobs in the cell. And this helps us predict the function of a newly discovered gene or protein. Okay, so if you have a new gene or protein, you can compare it to other proteins that are already known, and if you find a good match with a high sequence identity percentage, whatever, then that means that that protein probably does the same job as that one. Okay, fair enough. Right, then this one. Okay, hidden Markov model. Let's look at what that is. Okay, hidden Markov models are powerful tools in bioinformatics used for understanding sequences. Okay, the ability to analyze sequences where the underlying process generating the sequence isn't directly observable. Okay, what does that mean? Imagine flipping a coin, but you don't see which coin is used. Instead, you only see the sequence of heads and tails. heads and tails. Okay, so you just have the data on what was the coin flips, right? HMs try to guess which coin or state was being used at each flip. Okay, it assumes that the observed sequence is generated by a series of hidden states. An HMM is a tool that presumes that the observed sequence is generated by a series of hidden states. Each state has a probability of emitting certain symbols and a probability of transitioning to another state. By knowing these probabilities, you can infer the most likely. Okay, I have no idea what's going on here. So let's go a little deeper. Right, hidden states. Right, let's go a little deeper into that. Okay, internal mental. Okay, I think this is what it's referred to. Okay, let's hidden Markov model, right? Ah, okay. So a powerful tool used to understand sequences of events where the underlying process is hidden. Okay, it's like predicting the weather, trying based on what, guessing the weather based on what people are wearing. Oh, the weather is hidden, but the people's clothes are observations. Okay, makes sense. HMMs help us model this. They assume the system is in a certain state. Each state has a probability of transitioning to another state. Each state also has a probability of producing a specific observation. Sunny weather might make people wear shorts. Okay. Okay. So based on the current state, they can guess what was the previous state, like, or based on a few states, they can guess what was this before that and then this before that, right? Okay, fair enough. Now let's go back to HMM search. I guess this is familiar to me now in a way. Like, I kind of have a sense of what's going on. Now let's go into HMM search. Okay, it's a powerful tool to find related protein sequences. Instead of searching for a specific sequence, it searches for a statistical pattern learned from a family of related protein sequences. The concept behind HMM search relies on hidden Markov models. Think of an HMM as a sophisticated pattern detector. Okay, sequence. I see what you mean. Okay, so this is the key paragraph, right? HMM search uses this model to score how well a new protein sequence fits the family pattern, and higher score suggests that the protein is likely a member of that family even if its sequence isn't perfectly identical to known members. Okay, so we were looking at the templates in RMSD, right? Uh, okay, so this dataset excludes proteins with a template identified by HMM search from the training set. So they found templates using HMM search. Okay, so the reason they use HMM search is to find templates for the proteins that were in the training set. Oh, okay. So this is to they did this to further reduce the chances of similar proteins. So they really wanted to make sure that the model is actually good. So this shows very good level of like self awareness and like trying to reduce the bias. So this is good science there, right? They're trying to exclude if you've already trained on a protein in the training set which is very similar to what you find in the validation set, then you're not really you haven't really learned that much, right? If you have a template. So they find um, proteins in the validation set that are similar, that have a similar template to the ones in the training set, and they remove those, right, to focus on really novel proteins in a way. That's why this data set, this is worth mentioning, right? Overall median is 1.5. Okay, though this protein will be highly sensitive to domain packing and domain accuracy. Okay, domain packing and domain was like basically um, I don't know, is expected for some chains with uncertain packing or packing errors. Yeah, so basically they're saying that depending on, I don't know, highly sensitive to domain packing. I want to know what uncertain packing or packing errors mean here. Okay, let's look at domain packing. I already know, like, I kind of already went over this last time, which is the spatial arrangement of protein domains. Domains are like sub blocks within the protein structure that have specific functions, right? So improper packing results in higher RMSD values, indicating structural differences. Let's look at packing errors. Packing errors in protein structures are like mistakes in a perfectly arranged box of toys. Okay, so only okay. Sometimes when the scientist determines the structure of a protein, sometimes the arrangement of its atoms is not quite perfect. These imperfections, called packing errors, can arise due to limitations in the experimental techniques used to determine the structure. Okay, so basically this is like bad data, like errors in the data itself that you're trying to use. Hmm. Okay, so basically, like, if the data is not clean, if you don't know exactly what's going on, then sometimes you can have a higher, because you're comparing the position, the prediction to a truth, a ground truth, and that ground truth comes from some experimental data on that protein, right? And so a high RMSD is expected when you have packing errors, when the underlying data is bad or like has some errors in it. Of course, the predictions, even if their predictions are good, they might not match perfectly with the source data, right? They might be more accurate than the source data, in fact. So you never know. Okay. Next, we have the correlation between backbone accuracy and sidechain accuracy. Mm, this is B. B. Okay. correct. Okay, IDDTCA. I think that was we did went over this. Local distance different. Yeah, so we went over this last time, right? The local distance difference text. It's a metric. Let's quickly look at a metric assessing the accuracy of a protein structure. Yeah, this is like how much is the difference between the prediction and the result, right? And then fraction of correct x1 row timers. Okay, the higher this score, the more accurate, right? So this is more accurate, and so this is like what fraction of row tamers. Okay, let's see what that means. Filtered to structures with any observed side chains and resolution better than filtered to structures with any observable observed side chains and resolution better than 2.5 angstrom. Okay, out of basically 5,000 protein chains filtered by to B factor. A row timer. Okay, let's understand what a row timer is, right? So there's row timer, there's B angle, B factor, torsion angle, side chain. We know what side chains are, right? These are variable groups attached to the alpha carbon in an amino acid that give it more properties, basically, right? So we know that. The let's look at what row timers are. Imagine a protein is like a buried necklace, makes sense. Each bead is an amino acid and the string holding them together is a protein backbone, okay? Each bead has a side chain sticking out. A row timer refers to the different shapes that side chain can adopt by rotating around the bonds connecting it. Oh, okay, okay. So sometimes you have a molecule to molecule bond, right? And the bond, like this whole atom structure can rotate around that bond, that electrostatic bond, right? I mean, you have two magnets. You can always rotate the magnet, right? And based on the rotation, the structure will change. So the row timer is the different shapes that can be. Okay, think of it like your arm. Your shoulder is fixed to your body, but your arm can rotate. Different arm positions are like different row timers. Okay. The specific arrangement of the row tamers influences how the protein interacts with other molecules. Yeah, makes sense, right? If my arm is in a particular position, it will interact with the environment in a different way than if my arm was in a different position, right? Yeah, and this makes sense. The most likely row tamer traits. Okay, so row tamers, I'm familiar now. Side chains, I'm pretty comfortable in a way. Guess low. Okay, B factor. Let's look at what B factor is. Okay. Imagine you're taking a picture of a group of friends. Some are standing perfectly still, sharp and clear. Others are fidgeting. Okay. Moving during the photo, making them appear blurry. Okay. The B factor is like that blurriness. So it's a measure of how much an atom wiggles around its average position. A high B factor means the atom is moving a lot, which means uncertainty. A low B factor means it's very defined. Makes sense. Why is it important? Okay. B factors help us understand protein dynamics. Regions with high B factors might be more flexible and involved in binding or catalysis. The more blurry a region is, the more flexible it might be in binding, or the data might be less accurate. Okay. So yeah, so they were filtered to B factor. They're trying to filter it to the data to proteins which are tend to be more stable, less than 30 angstrom squares, basically, right? Um, And the predicted torsion angle. We went through torsion angle. Okay, familiar. Torsion angle. Let's see. A torsion angle is like a twist measurement at each of those links. It's the angle you see when you're looking down the bond connecting two amino acids. Okay, these are basically the torsion angles. A rotamer is classified as correct if the predicted torsion angle is within 40 degrees of each other. Okay, so basically this is like how they define if their own prediction is right, because you can't have a perfectly correct prediction, right? So if the protein is quite stable, if the underlying protein, the side chain is quite stable, right, which is like less than 30 angstrom b factor, b beta factor, whatever, then the way they decide if their model is correct or not is if it's within 40 degrees of the torsion angle, right, of the actual data. So they're allowing for a 40-degree variation as proof of accuracy. Okay, I don't know how they chose this, but up to, like, I mean, I'm not familiar enough with this field to figure out what's happening here, right? Okay, each point aggregates a range of IDDT. What was that again? LDDT, local distance difference text, right? I don't know how do you pronounce like this shorthand. The bin size of 2 units above. Okay, a bin size. Let's look at what that is. Interesting. What happened there? Let me just quickly check. What the heck is going on? Hmm. Okay, something's wrong here, and I have no idea why. Let's min size. Let me. Huh. And there is an error in the tool, and I have no idea what's going on. Hmm. I have no idea what's going on, but I'm gonna have to stop this live stream probably and figure out what's wrong.